So first I would like to thank the organizers for their very nice invitation and for organizing the workshop even in those conditions. That's really nice to have an in-presence hybrid workshop. So today I will present a model for the tissue growth for embryo elongation. That will take into account the fact that we have multiple tissues in the embryo, and also the fact that there is viscosity of the embryo. So this is a joint work with Bertrand Benazeraf, who is an embryologist in Toulouse, with Pierre in Toulouse, with Sophie Hesch, who is now in Paris, at l'ILL, and with Michel Romanos, who is a PhD student supervised by myself and by Bertrand Benazeraf. So let's start with the embryo elongation. So at the very beginning of the life of the embryo, it is like a round or symmetrically like shaped, and suddenly there is a symmetry breaking, and it starts to elongate in one direction, and this elongation is very radical, really decided along one axis in one direction. And this elongation is actually quite complicated. And in particular, one first thing that we see is that we have multiple tissues. So let me present you these tissues. Here you have a picture taken by the group of Bertrand Benazeraf in his lab. So this is an embryo of a bird. You have the head in the top, the quail in the bottom. What you see in the very thin dark line is a tissue called the neural tube. It will form notably the nervous system, the nerves and so on. And what you see around it is the gray part, is the PSM, the mesoderm, which will form some organs like the muscles and so on. In the drawing, the PSM is in red here. In green, you have the neural tube. So it's really in the central. The neural tube is surrounded by the PSM. Just so you know, these little dots or runs that you see here are prismites. So these are the future vertebrae. We'll be particularly interested in this bottom part that is very active during the elongation. It's the one that elongates the most. Okay, so I will first show you a picture of what we see. Works? Okay, good. So that's the video is, the photo was extracted from a similar video. And you see, well, the vertebra forming here, or the prismites, the elongation, and at the end you even see the heart forming and starting to beat. And at the bottom, I did not mention, but there is this progenitor zone that is very, very active. Okay, so when we look at it, we definitely see that the tissues have different colors, so or different shades of gray. So they seem very different in terms of density or maybe other properties. And they are in contact with each other also. And something, I don't know if you can see on the video, but there is a very complex choreography at the cellular level. So you can see all these little dots are actually cells, and you can actually track them with adapted, I don't know if you can with your eyes, but with adapted tools we can. And what we see is that there are very complex movements. So you could imagine, I don't know, that since everything is elongating, maybe, I don't know, the cells are going in the direction of the elongation and everything is fine. Not at all, of course, there is a lot of chaotic movement, a lot of diffusion. The cells are migrating in random directions, but also even if you like get rid of this, clean this chaotic movement, you see that there are also more interesting movements like some vortices that form. So, for example, if you try to follow, where is my? Yeah. If you try to follow around here, you see that it moves more or less like that. And so there seems to be a kind of order behind all this. So that's what we want to study. That's actually the quest of Bertrand, to study how the tissues actually co-evolve. Do they interact? Do they evolve differently? And also how the choreography of the cells is related to the movement of the tissues. Okay. In particular, what was actually, I mean, what we see is we can quantify, and it was the object of the research of Bertrand, is that we can see that the tissues have different elongation speed. So by that I mean that if I define more or less a bottom and a top of the tissue, I will get actually different speeds, and also differential growth in terms of. So this is more related to the cell division. And also he showed that you can quantify this turbulent or rotational movement. So I will show you a small movie on that. So this is a ventral view of the embryo. Yes. Okay, it starts around here. So it's just a small region of the embryo. The top is in this direction, the tail is in that direction. In the middle you have the axis. This is closer to the progenitor zone, and what is more red is the PSM. The colors indicate the clockwise or counterclockwise, the rotation actually of the cells. So the cells are tracked, and then it is computed in which direction they seem to turn. And you see that it seems quite ordered. Basically, what you can imagine is that cells, they go away from here, the progenitor zone, they go to the PSM, but also to the neural tube. So you have movements that create these rotations, and here it creates turbulences in the PSM.
Just one question: it's rotation around what? Around the center of mass?
No, it's local. So it's like if you compute the velocity of the velocity field, you compute the velocity field, and from that you look at the rotational. Okay, so now, of course, this raises a few questions. So the first one will be how the different natures of the tissues will affect our dynamics. So we have different tissues. They may not have the same properties. So we know that they don't grow at the same speed. They may have also different mechanical and biological properties. These tissues, they might also interact. For example, the neural tube is really in the middle, surrounded by the PSM, so we might imagine that some pressure happens. And there might be friction or not. Maybe one tissue is actually more active and is pulling the other one, this kind of questions. And the other question is how to explain the turbulences at a tissue level, and have these turbulences a role in the elongation. So to answer this question, we want to do a mathematical model. There are of course many ingredients we could put in this model, and I cite a few of them. So there is a proliferation, so the growth of the cells. There's also the fact that the cell die. There is a migration of the cells, migration that is can be a bit random, like diffusion, but also this migration can be due to congestion, since there is a lot of growth at this moment of the life of the embryo. Well, there are many cells appearing, so it creates congestion, which can impact the migration. The interaction between cells, especially cells of different tissues, and also interactions that we will not look at with the extracellular matrix, chemical signals that we'll not consider. Somehow we want to test how much we can obtain with only, let's say, mechanical models or local. Somehow I did not put any chemotactic movement or any signal at a long scale. And well, there's also differentiation, since so the progenitor zone is feeding each one of the tissue, and there's a big question on are the cells differentiating before moving, and then they move to the right tissue, or are they actually moving, and then once they are in the right tissue, they differentiate according to the tissue. This we don't really know, so we did not look at it, because we don't have enough data, and that was not the main point of this project. So, since we're looking at the tissues, we will do a mathematical model at the scale of the tissues. So let's say macroscopic model, and more precisely a mechanical model that can take into account all these effects. Can I ask you at what time I started?
I think you have plenty of time.
Yeah,
you started precisely at quarter.
Okay, thanks. Okay, so I will first, recall more classical models for tissue growth. And in particular, when there is one single tissue, one way to represent the growth of this single tissue is to look at the evolution of the cell density of this tissue. So we call N of Tx the cell density, T is the time, X is the space. V of Tx will be the velocity of my tissue. And I have this first equation that is just the equation of evolution of N. There is migration with the velocity V of the tissue, and some growth with rate G of P. And for the velocity, I take as a simple example for the moment what is called Darcy's law. So P here will be the pressure inside the tissue. And when I say that V is minus gradient P, it's a simple way to express the fact that we assume that the cells will go from high regions, places of high pressure to places where the pressure is lower. Okay. And this pressure, in my case, I choose it singular, so I will take P of n is equal to epsilon n over 1 minus n. And this means that, well, first there is no pressure when there is no density, but also if I take, I mean, it forces my density to stay between 0 and 1. So somehow I have a maximum capacity, maximum density, n equals 1. So I took 1 arbitrarily, but of course you can change this number. And this means that I don't have any overlapping. So at some point, maybe, I don't know, I can consider that I have a hard sphere kernels or something like that, but I cannot go above n equal 1. And the last thing I want to mention is that here I suppose that the growth also depends on the pressure, which means that so I would typically take G decreasing. This means that the cells will tend to slower their growth when there is too much congestion. Okay, some references. So actually it was first introduced with a power pressure law. So pressure law that can be a bit different. And then this singular pressure law was proposed to model the non-overlapping. Okay, now let's go back to this Darcy's law for the velocity. So this is a very simple, This is a first simple model. Now in our case, I told you that our tissues are viscous. And not only they're viscous, but actually we have many tissues and they may not have the same viscosities. So you can imagine, for example, the neural tube will form the nerves, the PSM will form the muscles, so you can imagine they don't have the same texture intuitively. So it seems very important to take into account viscosity, also to be able to take into account the fact that they have different viscosities. So for that, we use the Brinkman form of Darcy's law. It writes minus beta Laplacian V plus V equals minus gradient P. So it's kind of a regularized version of the previous one, with beta being the viscosity. So viscosity. The second thing with the Brinkman form of Darcy's law is that it allows a non-zero rotational. Actually, if you take V as a gradient, well, rotational of V would be rotational of gradient would be 0. Now, if you take V not as a gradient, if you take it as the solution of the Brinkman law, if you're in a bounded domain and you take clever boundary conditions, then you might have a non-zero rotational. So this is just a simple representation of the solution, well, of the curl of the solution of the Darcy on left and Dirichlet and the Brinkman on right. What you see in black here are the arrows. So here you have Darcy, are the arrows of V. We took for a pressure something that is 0 outside this round and that form a bump in the middle. Same thing here. And so you have arrows in the middle. Here you have arrows a bit everywhere. And so that's V. And the colors indicate the rotational. As you can see here, the rotational is 0 everywhere. And here you see that you have positive or negative values for the rotational. Yes?
How is it possible that the curl is not 0? Because the curl of a gradient is 0. V is that a gradient?
So here, V is a gradient. Here, V is not necessarily a gradient. This depends on the boundary condition we choose. But here, we took a Dirichlet boundary condition, and V is not a gradient. So now, let me comment on the fact that in some cases, we can take V as a gradient, indeed, even with Brinkman law. And actually, some very nice people have studied this case. And so, especially in the whole domain, you can take V as the gradient of minus a potential W. And in this case, if W is the solution of this equation, of this elliptic equation, then you see that when you take the gradient, you will recover exactly the Brinkman form. So in this case, what is nice with this model is that you indeed have viscosity, But what doesn't work for us is that the rotational will be 0. So that's why we cannot allow this form. And also we need to be in a bounded domain. Okay, so after this comment, just to simplify, I go back to Darcy's law. So I have this description, one equation for n and one equation for v with my singular pressure. And I propose also, I mentioned also another description, which is a geometrical description of the same situation. More precisely, omega will be the domain where I have tissue. So instead of describing the density of the tissue everywhere, I just consider in a simplified model that I have tissue somewhere and in the domain and no tissue around it, and somehow my domain of tissue is how to say, fully occupied. And this comes, I don't know if I can use that actually. No. This comes with some equations on the pressure. So I solve the equation for the pressure and the equation of the pressure. So the pressure will give me the velocity of the boundary of the domain. So that's another description which is way more economical and that will interest us also. And one way to link these two models is to compute the incompressible limit. So in our case, we took this singular pressure law. It consists in taking epsilon to 0. So first you see that this will force n equals to 1 whenever p is non-zero. From the p of n is epsilon n over 1 minus n. And yeah, then the velocity of the tissue is linked to gradient p here in the case of Darcy. And in this case, you see that the velocity of the boundary, which is the velocity of the tissue at the boundary, is also linked to gradient p. Okay, so and well, I'm not sure I cited everyone, but many people worked on incompressible limits from this type of model to this type of a model for one species or two species, different laws for the velocity, singular pressure or not, and also I don't know, adding nutrients and other variants. Okay, now I will go to the case of the case that interests us for the embryo, where I have multiple tissues. So now I have let's say two tissues to simplify. I will have one equation and one system of equation for each of the tissues. So one now Ni will be the density of cells for tissue I. Vi will be the velocity of tissue I, and Pi will be the pressure felt by tissue I. This should be a Pi, I'm sorry. So you have again migration with velocity Vi and some growth. Again, a Brinkman form of Darcy's law to have the viscosity. The viscosity can be tissue dependent, beta I. And now the question is, what kind of law do we take for the pressure? So the pressure will will insert a coupling between N1 and N2, since it will depend on the two densities. So that's where you encode really the interaction between the two different tissues. In the first model, we can just adapt the previous case where we had only one species, and we say that our pressure P is the same for N1 and N2, to simplify, and it is the congestion pressure, the same as before, singular, epsilon n over 1 minus n, but now n is the sum of the two densities, so it's the total density of tissue. So this model, I will call it the VM, the viscous model. Well, because, or two-tissue viscous model, because I have two tissues and I take into account the viscosity. And in this case, what's interesting is that I have the segregation property for this type of model. So the segregation property is the fact that if I take initially segregated densities, then they remain segregated for all times. That is, if n1, n2 is equal to 0 at time 0, then I should have n1 and 2 equals to 0 for all time t positive. That is, I cannot have n1 positive and n2 positive at the same time. Okay. Actually, for us, we thought that this was not enough, and we wanted to try to impose segregation. The thing is that in our case, the tissues that we will consider will be the neural tube and the PSM. So they are almost segregated, let's say. I mean, if you look closer, of course, everything is heterogeneous, but they appear as mainly segregated. Like you can analyze what type of cells, what type of proteins appear, what types of cells are in the different tissues, and we can consider that globally they are different in the neural tube and in the PSM. But they are in a contact with this progenitor zone that is completely mixed. And since we don't really know how this migration and separation arises, we wanted to force it. And for this, we consider what we call the repulsion pressure, Qm, which is a function of n1 and 2. So Qm of n1 and 2 will be m over n minus 1, 1 plus n1 and 2 power n minus 1 minus 1. And you see that when m will be large, you will take n1 and 2 to 0. So this forces somehow n1 and 2 to be close to 0, which means that you force segregation. And we adapt the pressure accordingly. So now the pressure of the tissue I, the pressure felt by tissue I, will be the congestion pressure. We keep it with a circular pressure law here. But also the term given by the repulsion pressure. So for P1, it will be N2QM. I add the N2 because I want this to be in the right direction. I want somehow to enforce I mean, I don't want only to segregate locally, but I want to form like packs. And I end up with this model. So what's in this model? Again, you have an equation for n1 and 2, the velocities v1, v2 with the Brinkman law, or Darcy form of Brinkman law, the pressure with congestion, and also with segregation, with repulsion. And we added these quite ugly fourth order terms that are necessary to avoid bubble effects. So the bubble effect is when you have initially mixed densities, and you would hope that they would form two connex, let's say in 1D, two connex sets. And but if it's mixed, you may have instead of two big sets, you may have little bubbles like this. So alternate a very small portion of N1, then N2, N1, N2, and you want to avoid that. And alpha, the presence of alpha, was shown to avoid this type of effect. So in the paper of Chartok, Degon, Eschenbach. Yeah, I think I can show you some simulations now for this system. Okay, so here, this simulation in MATLAB. In the middle, you have the neural tube. What you see, the colors are densities. Here you have PSM, and well, you see that it evolves in in the direction of the axis, so everything goes well. Now, if you look at the fluxes, well, you see that some rotation seem to happen. And actually, I don't have the I don't have the representation of the rotationals, but you can also compute the rotational simply and see that you can indeed have some rotational, and it's a non-zero rotational. So that's one thing we wanted to obtain. Okay, and now what we want to do is to compute the incompressible limit of this model. So in this case, the incompressible limit will be not only to have epsilon goes to 0, but also we want segregation to be enforced, and we will take m goes to infinity. And then take also alpha equals to 0, since we expect that regularization should not be needed anymore. The first thing we can do is, and here I need to do a little comment. This incompressible limit that I will show you, I will do it completely formally. When it comes to rigorous, the rigorous limit, the closer that is known at the rigorous level is the case where there is no q, there is no alpha. So since you don't put q, you don't need to put the alpha, to put alpha equals 0. And you have the Brinkman law, but in a gradient form. At the moment, so even without the q, even without the alpha, for a velocity that is not in a gradient form, we miss a maximum principle, so it's not an easy adaptation from the gradient form, and even in this case, it's not an easy task. So for the case where you add this singular segregation and you add these 4 order terms, it's even, I mean, it's way, it will be in the second step. I mean, it's way more complicated. So I will show only formal computations. The first thing that we can do is that in the four equation, if you assume that everything converges towards something that is bounded and so on, the 4 equations, you can rewrite them basically without the alpha term at the limit. So you get, well, an equation for in1 at the limit, that I call an in1 infinity, with the growth and the migration, same thing for n2. You get the Brinkman law at the limit, and the pressure I defined inside each tissue thanks to P and Q. The second thing you see quite immediately is that that's the reason why you're taking the incompressible limit. When epsilon tends to 0, whenever P is non-zero, you will get n equals 1. And also, you have this segregation because if you want this to have a finite limit, you need n1 and 2 equals to 0. So that's the second thing you see. You have p1 minus m equals to 0. So you did suggest to define the domain where n equals 1, where n is the total density, and 1 plus n2 at the limit. And we have full segregation of the species. So now you want to divide omega into 2 domains. So in a nice situation, you can have, for example, a first domain omega 1, where n1 is 1 and n2 0, and another omega 2. And the velocity of your free boundaries are simply inherited from the velocities v1 and v2. And well, nicely, your velocity coincide at the zone of contact. So you have a nice description. The only thing is that you need to close the system, and to close the system, you need to find equations on P and Q. To find the equation on P, so P, I recall you, it depends on the total density, n1 plus n2. So before limit, you can write an equation for n1 plus n2, and then it gives you an equation for the pressure before limit. You can pass to the limit, and you end up with an equation for P. So this equation will look like that in the end. Oops. So this was already known for other systems. Now what's more complicated is that you also need an equation for Q, and it's actually way more complicated to get one. And well, actually we didn't really manage to get one directly, but what we do is that we look at n1q on one side, I call this quantity u1, n2q on the other side, and same thing, I can get equations before limit for these quantities, and I can pass to the limit into these equations, and I get these final equations that are, well, what they are. And what is nice with these equations in the end is that so they depend on Q on the right. But basically, they will U1 will give me some Q when N1 is positive, and same thing for U2. And I don't know anything on Q whenever I don't have any densities, when N1 equals N2 equals 0, but this I don't need it, because Q does not appear anywhere outside of the domain, omega 1 union omega 2. Okay, so that's my incompressible limit. Now let me comment a bit on what we obtain. The first thing that we see is that at the limit we have this full segregation, n1 times n2 equals 0, but still some segregation pressure seems to persist. We have an equation for this segregation pressure, and well, nothing tells us that it should be 0. On one hand, when we take Q equals to 0 at the limit, we give the special solution, so Q equals 0 is an admissible solution, and we can study the system in this case. On the other hand, we did numerical simulation, and the fact that Q positive at the limit is possible. So when I say numerical simulation, I did mean that we performed the limit. We tried to perform the limit numerically and see whether Q converges to something or not. And it seems here that we can obtain a finite non-zero limit, and we called it a ghost effect, as in kinetic theory. So how to interpret this? Maybe one way to understand this is that even at the limit, segregation can still produce some forces and still affect the dynamics. For example, okay, what does it mean that Q infinity is non-zero? It means that when you pass to the limit, you segregate your tissues, but you don't segregate fast enough. So the Q of N1 and 2, the first order terms, for example, will keep here in the equations, and basically you have a 0 at power infinity. 0 is your N1 and 2, and yeah, it's indeterminate. So somehow this means that you don't segregate fast enough. So this means that in the end, you may have some residual of densities, some residual of tissue 2 in tissue 1. And these residuals still produce forces, even if we don't see them at the macroscopic level. So we don't see the density, but we might see how they affect the dynamics. And well, the other is the contrary. It means that the pressure Q vanishes, so that there are no microscopic residuals, or they're too small, and they just don't affect the dynamics. Okay, and yeah, this is just a comment that, so we have, I showed you two models with viscosity. One without the enforced segregation, so no Q and alpha equals 0, and one where I added the force. And we can actually check that, so the two systems, they coincide if you take initially segregated densities, and if you take alpha equals 0 for the ESVM. Because then initially for the ESVM, Q will be 0. And then it should not, I mean, then you are equivalent to VM at time 0, and you keep being equivalent. So when everything is fully segregated, you have a coincidation. These two systems coincide. On one thing. On the other hand, we're happy to see that in the case where we take initial data that also don't mix, and you take Q initial, which is equal to 0 in this in the limit of the ESVM, then you recover exactly the system obtained as the limit of the VM. This one is yeah, quite interesting because you have this ghost effect, this segregation force that remains in general. Okay, now I'll give just a small hints of analysis of the limit of the solution. We will work in a special case, in a simplified, not special actually, a simplified case. Which is linear. So to simplify, I take a growth function that is linear and strictly decreasing. I look at the stationary case. So when I say that I look at the stationary case, so at the limit, since I have moving domains omega 1 and omega 2, I just fix these bounded domains. So I'm not at all looking at the evolution of the domains anymore. I'm rather looking at the flux and trying to solve the stationary. And also, which is a big simplification, I consider that Q is given. So I will just erase the equation of Q for the moment and yeah, look at the case where Q is given. For example, we can think of Q equals 0 if we are in the LVM case. And I add homogeneous Dirichlet boundary conditions for the velocities. So in this framework, I can rewrite the system as an elliptic system for V1 and V2 in a bounded domain theta. And under some assumption on the parameters, I can prove the well-posedness in H1, so I have my velocities that are continuous, which is nice for my domain. I mean, if I want to compute the velocity of the domain, it's better that the velocities, I mean, doesn't have jumps at the boundary. And I can show regularity of the variables, the velocities, inside each of the subdomains. And also I can rewrite the problem as a transmission problem. So I wrote here the transmission problem in the case where Q equals 0. I'm not asking you to really read all the equations. I'm just saying that basically I have an elliptic equations for V1 and V2 inside each of the subdomain, omega 1, omega 2, and omega t. And then I have many transmission conditions on the boundaries. Okay, I do have to. Okay, I'll be. Okay. And just one thing is that my transmission may copy a jump, and actually there are jumps at the boundary. We could show for q equals 0 that there is a pressure jump. So that's interesting qualitatively. And this pressure jump was, we recovered this from the case where this is in a gradient form. So that's consistent. And so to conclude, we developed a model viscous multi-tissue with enforced segregation. We derived from it a geometrical model. And what's interesting is that we observed the persistence of a segregation force. And we rewrote this model as a transmission problem. And from this, we have one qualitative property, that is the pressure jump at the frontier. Of course, there is still some work. We are working on elucidating this ghost effect, in particular linking it with initial condition at the before limit. Comparison with biological data with Bertrand Benazeraf, and well, this rigorous limit, not for this full alpha, I mean Q non-zero, but even when there is no alpha and Q, but with the generic Brinkman law, and some link with some microscopic models, as was proposed by, I mean, in a previous publication, Romanos et al. In eLife, where, so it's in we propose a microscopic model that can also explain the dynamics of the tissue, and it would be really interesting to link the two models. I thank you for your attention.
Thank you for your talk. I referring to the model with the fourth order operators. Yes, I was wondering whether, how can you prove, if you need to prove it, but I think so, any sign property for N1 and N2?
Yeah, so your question completely makes sense, I agree. We do numerical tests, basically.
Yeah, no, I understood that. No, I was wondering because...
Yeah, how can we prove it?
Yes, maybe you need some modification in order to get a rigorous result, say.
Yeah, I think so. But at the moment, I'm not trying to get rigorous results on this, this model with the alpha. Basically, already the model without the alpha is yeah, okay. Some work. But I completely agree that it would mean it would need some work on positivity.
And then in the big system of equation with U1, for U1, U2, you get here, where do you see the trace of the Canelliard terms? Of the fourth terms, do you see it there somewhere?
We take the alpha equals 0 at the end, so we don't see it anymore. In the, you mean the fourth order terms?
Yeah. Yes, alpha equals 0, but it could stay something out of, I don't know.
Yeah, I see what you mean.
But you don't see it at all.
No.
So this model is in your big picture with 4 arrows. Where is it?
It's this one. But I would rewrite it inside each subdomain.
Other questions?
Just a question about this ghost effect and its link to the connected components of your subdomains. The number of connected components, because I still don't understand how you control when alpha is 0, how you control the number of connected components of your subdomains.
Well,
why don't you get infinite number of components with little components popping out everywhere?
Yeah, I see what you mean.
Would that be marked by this ghost effect? That's the question.
Yeah, I think I never really related both questions. I don't know. Could be interesting to see. It's true that since we're working at the formal level, we don't know whether we can avoid, this effect. We just assume that it works because in the end we want a geometrical domain with a finite number of components, and also numerically it seems to work.
Without surface tension, I don't see how you can ensure that. With surface tension, everything I think is nice and natural, but without it, I really don't see how you can enforce that. And maybe if you have alpha different from 0, then there's no ghost effect. Right?
If you have alpha different from 0, we take alpha to 0 because we have to. I don't know what happens if we keep the alpha positive at the limit. The thing is that we have full segregation, so if we keep regularizing our densities, not sure. I imagine it breaks everything.
Okay, thank you. We can talk later. You're staying around.
Yes.
And the question is there any relevant way to link alpha and m when going to the limit?
No, of course, that's... So is there a way to link them?
Yeah, if there is a way to do that.
We can link them, but that makes appear another quantity that we don't know. So we believe that there should be there should be some link, but we have not elucidated it yet.
And then it says a reply, because alpha equal to 0 first means bubble.
Alpha equal to 0 means, sorry?
Because alpha equal to 0 first means bubble.
It can...
Bubble effect.
Yeah, it can create bubbles, yeah. Yes, indeed.
Yeah, I think that was the origin of the question.
Okay.
But I think you answered the question. That's okay. Thanks.
Okay, no more questions. We can thank Ariane again.
Thank you.